Hi, good evening, Dr. Miller. Thank you so much for being part of the Merck's BioLuminescence event. I want to ask you the first question regarding the rapid microbiological methods, as in when the organizations intend to employ these technologies, what should they look for?
Yes, so rapid methods have gained in popularity over the last few years. A lot of this is due to the increase in manufacturing of gene and cell therapy products, which really do require rapid methods. But companies who wish to implement rapid methods should first understand where the opportunities are for moving away from conventional or compendial methods to the more rapid and alternative methods, and then develop a strategy for implementation, which includes validating the system to demonstrate that it is not worse than the conventional method, and perform some method suitability studies as well. At the end of the day, the rapid methods should be shown to be suitable for their intended use and give you good results much faster than what we currently use today.
Absolutely. So when you talk about the compendium and the advancements, would you like to highlight something there?
Yes, well, over the years, a number of the pharmacopoeias have added chapters to provide guidance on how to validate and implement rapid methods.
Right.
The current work that's being done by the USP includes the development of test chapters specific for 3 different types of rapid methods. There are others as well, but the 3 that I'd like to highlight include respiration, and ATP bioluminescence, as well as solid phase cytometry. And the purpose of these chapters is to make it easier for the industry to implement those scientific principles. Now, take ATP bioluminescence, which is relevant to this conference today. The end user would utilize that particular chapter when it is published to just perform method suitability, and then they can immediately start using the system. And this is because ATP bioluminescence has been used for many, many years, so it is considered to be a validated method. This is the intent of the USP chapters going forward.
That's wonderful to know. Also, coming back to the point that you raised in the first question about cell and gene therapy, that's most trending these days. When we talk about the rapid microbiological methods in application here, what do you think would be the challenges globally or from an India perspective in that?
Yes, so cell and gene therapy, or ATMPs, this is the fastest growing segment of pharmaceuticals, and it's a much needed medicine for individuals, especially those who are battling cancer. So these oncology patients, many of these types of modalities are ones in which they provide their own blood cells, which are then reimagined into fighting cells that are specifically going to look for their cancer cells. So they provide the blood samples. The manufacturer will change those blood cells to recognize the cancer, gets reinfused back into the patient. The issue here is that many of these medicines have a short shelf life, and a 14-day compendial sterility test just doesn't work anymore. So in order to release the test and be comfortable that each of the batches are safe for use, a rapid sterility test is certainly needed.
Okay.
So that is one of the biggest pushes for ATMPs, cell and gene therapy, and the use of a rapid method for sterility testing. The other importance here is, these patients really need to be treated with these medicines as quickly as possible. There is an immediate medical need, another reason to have the release of those products as quickly as possible.
And do you foresee any regulatory challenges around this?
No, actually I don't. The regulators around the world understand that these types of medicines are needed as quickly as possible, that the scientific principles around rapid methods actually work, and many of the regulatory agencies have provided approvals for rapid methods for release of cell and gene therapies. So there's no challenge from a regulatory standpoint. It's just up to the industry to get on board and develop their implementation plans.
Absolutely. Seeking light on one more aspect is around automation when we talk about the microbiology QC related methods. How relevant is the concept of automation now?
Yes, it is quite relevant. The more testing that's done, the greater capacity that microbiology labs will need in order to run the tests, get the results, and get these products to the patients who need it the most. And there are a number of platforms now that automate the process of what technicians have been doing manually for hundreds of years. Okay? So automation is going to help in a number of ways. We can get more samples tested in a given time. There is less of an opportunity for errors in terms of data management and data integrity, because most of these systems are going to document the results and do that electronically, and for some systems to be able to promote the procedure themselves to the specific technician, so that each step of the procedure is actually followed through, and when those steps are done, these are recorded electronically. So very exciting time for automation.
Absolutely, without doubt, exciting times are ahead, and we look forward to more developments. Thank you so much, Dr. Miller.
Thank you.
Thank you.